Welcome everyone. Today I'll be solving question number 7 of the 2021 GATE Biotechnology paper. The question is that which one of the following statements is incorrect about hybridoma production? And there are four options given to you. The first option is that hybridoma cells can use hypoxanthine and thymidine. The second option is that DNA synthesis in myeloma cells is blocked by aminopterin. The third option is that hybridoma cells are made to produce polyclonal antibodies. And option D is that polyethylene glycol is used to fuse myeloma cells to B cells. So let's understand a few basics about this question first. And we can start with the difference between monoclonal antibodies and polyclonal antibodies. So polyclonal antibodies are antibodies produced by many clones of B lymphocytes to bind to a variety of epitopes, while monoclonal antibodies are identical when produced by one type of T cell and all are clones of a single parent cell. The second thing that we need to understand is the hybridoma technology, which is used to produce these monoclonal antibodies. So the first step of the hybridoma technology is the immunization of a mice against the antigen against which you want the monoclonal antibodies to be formed. The mice will produce many types of B cells against which antibodies can be produced against this antigen. You will extract these B cells and fuse them to myeloma cells using PEG or polyethylene glycol. And you will end up with a mixture of cells which you will pass through the hatch selection media. And because of this hatch selection media, only hybridoma cells will survive. Once you get the hybridoma cells, you can expand the desirable hybridomas and extract the monoclonal antibody. The hatch selection media that is used in this case is named after its components, which is hypoxanthine, aminopterin, and thymidine. Hypoxanthine and thymidine are precursors to nucleosides, while aminopterin is the inhibitor of the HTPRT gene. So in this media, what happens is that only hybridoma cells can grow in the hatch medium. Any other cells will not grow in this. So using this medium, you can isolate only hybridoma cells. You can see a flow chart of the hybridoma technology. So once the cells are fused using PEG or polyethylene glycol, you get four types of cells: fused B cells, hybridoma cells, fused myeloma cells, and unfused cells. Now, these four types of cells, when grown in hatch media, what happens is unfused cells, since the myeloma cells don't have the HTPRT gene, they will not survive in this media since they cannot use the hypoxanthine and thymidine. And the B cells don't have an immortal lifespan. So after a few cell divisions, they will die. Fused B cells will also have the HTPRT gene. They can use the hypoxanthine and thymidine, but like the unfused B cells, they will also not have a immortal lifespan. So they will also end up dying. So the only cells that are remaining are the hybridoma cells. So let's see the question again. So the option A, which was hybridoma cells can use hypoxanthine and thymidine, are correct, like we saw. DNA synthesis in myeloma cells is blocked by aminopterin, which is also correct. Hybridoma cells are made to produce polyclonal antibodies, which is wrong. And polyethylene glycol is used to fuse myeloma cells to B cells, which is also correct. So the incorrect option is option C, which is hybridoma cells are made to produce polyclonal antibodies. That is the answer for this question. I hope that was easy for you to understand. Thank you so much for listening.